Okay, with that, let's recap. So proteins can be seen as on the level of the coordinates, 3D, 2D is a simplification of the contacts or distances between any pair of residues. That is essentially containing all the information of the 3D structure. Projection upon 1D completely throws away that information, but it still has some information. Proteins in similar families have similar structures. This is a superposition of 50 different structures. And we can in fact find sequence similarities by pairwise sequence alignments. And in order to sequence align, we are not only matching the top sequence identity in terms of identity of letters, but we have substitution matrices, such as the Blossom scoring matrix here, and we compare the sequence using such a probability of exchange matrix against another sequence. Then the advanced way of sequence comparisons is the sequence profile, in which we take a single sequence and compare it against a profile from a family. And this profile for a family, at every single position, sequence position, so at every single residue, you have a certain probability of, in this particular case, we have 4 entirely conserved positions. They are in purple here, or 4 largely conserved positions. And that is where, if you bring in the sequence into this family, you really have to match that first. The one step further is the profile-profile comparison, where essentially we take one profile and align it against all the profiles in a database of all the families. With these methods, we get from very simple alignment techniques where we can align the 2 proteins that have 99% sequence identity, increasingly down to the twilight zone where really the signal is disappearing. A couple of the slides that I showed today are from Andreas Schafferhans. Let's begin with the background. So again, similar, the information for how the protein structure looks is in the sequence, and in principle, all the information about how the protein structure looks is entirely written in the sequence and in its environment, which you can imagine to be water in some sense. And in principle, what we want to do is take the sequence as input and get as 3D structure as output. The fact that the sequence contains the entire information led to the Nobel Prize for Christian Anfinsen in chemistry in '72. He established that you can simply take a sequence and unfold it and fold it and refold it, and all you need is the sequence and water. Let's just look at a structure here and look at what change in sequence leads to. So this is a particular protein structure. The protein is the oncogene KRAS. Oncogene means it's important for cancer. And so here are the beta strands, here the helices, the structures that the kind of typical symbols that you have seen before. So now what we see here in red and rainbow, we see 2 different structures, and these 2 here, they differ by 1 amino acid. Okay, so for these helices, and most of it, you see that the 2 structures are very similar. They differ in the binding site. Now this, in fact, this variant, the single sequence variant in this particular case, causes cancer, or has to do with cancer developing in the carrier. Here we get down to a level where we actually look at 2 proteins, both of them are human, essentially similar proteins. They're both KRAS-like. One is KRAS and one is RAS, so-called RAS. They have 85% sequence identity, so they are still very, very similar. And you see that, so one is this lime color and one is the green color. Essentially, they are very, very similar, right? Now we get down in terms of sequence identity. We get down to the level of fly. 28% sequence identity, so those 2 first, the lime and the green and the lime, are both human proteins. The orange one here is now a fly protein, but it's still very similar. Without having looked at many structures, you will immediately recognize that they look very, very similar. And in fact, so this is in some sense what we talk about on this side here, where it essentially is relatively simple. We can even go one further. In this particular case here, we can go in purple to a bacterium. And you see that even in the bacterium, this particular protein is still relatively well conserved, in the sense, of course, historically, thank you, Dimitri, in terms of evolution, it goes the other way around. Most likely it starts from the bacterium. Okay. Would it actually have been enough without the microphone for everybody in the room? Okay. I take it as a no. It started from bacteria in this particular case, so that is the origin, the purple structure, and the orange, lime, and green are evolutionarily later structures, but as you see, the structure is the same. Essentially the same, very, very similar. Same is not quite right. Now, when we look at the universe of all the structures, here's one figure from Christina Rengel. In fact, that was the first paper that ever showed a whole bunch of structures together on one slide. Is this one here from '97. And you see there are a variety of these structures. Comparative modeling now essentially is to somehow look at a sequence and see whether it maps to any of those. Not necessarily the ones on the whiteboard here, but the universe of all the structures. So map it into the 3D structure. How could you possibly do that? How could you map a sequence to a structure? I'm not entirely sure. So when you say we have to know the possible angles, do you mean that I take the sequence, I look at it, and I say this can form in that way and that can form in that way? Is that what you mean?
I mean, we have to know all combinations of...
Yes. Now you're getting, so he says that what we have to know is essentially all possible combination that a particular sequence can adopt. And now we are unfortunately in the combinatorial problem. So the combinatorial space that you have is extremely large. We do not know that. We will get a little bit back to the issue of how much through physics energy you can do, biophysical energies through molecular dynamics simulations, but ultimately we cannot do that. So today, this lecture is confined to the simplest way in which we can do that, which is take a sequence and map it to one of the known structures. So map it to one of the known structures. Every structure that you know has a sequence attached to it. So for every protein for which you know the structure, you of course know the sequence. How could that help you? Just think about this one here. So assume that I wanted, for the dark green KRAS, wanted to know how its structure looks, and I don't have neither the orange, nor the lime, nor the green. What I do have is the bacterial purple. What could I do? So purple is the only experimental structure that I have. In this particular case, I don't. Assume that would be the case. How could you predict the structure for the green? Yeah?
I could compare the sequences and then estimate with my probability how much percent they overlap. I could maybe estimate how it will look. If
yes.
It's the best match.
Yes. So for the people who cannot hear him, we could compare the sequences. So we have the purple sequence, we have the purple structure. I take the purple sequence and I compare it to the green sequence. I find the best superposition by pairwise or sequence profile alignment, and then I somehow infer the structure. And this is essentially the content of today's lecture. And I'm not going to tell you much more, just a little bit of additional details, but that essentially is exactly what comparative modeling is about. So I have a query, and that's the language here. The query is the one with which you start your search. As a query is easy for you. And you have a structure in the PDB. The PDB is the protein data bank. That's the database where you find all the known 3D structures. For that structure in the PDB, I have actually an experimental structure. And then I simply align them and I look up where for the corresponding residues, for the corresponding residues the structure looks, and essentially copy that coordinate. That's the simplest approach. Now, let's see how far we can reach with that. So how many proteins in today's universe? I said there are about 65 million proteins in the sequences known and about 100,000 structures. So how much can we do in this way? And again, the method I will explain in a little while later. So now I'm going to show you a pi. The whole pi is essentially the universe of all protein sequences. And it's an extrapolation that I show you. And in this extrapolation, the universe of all these protein sequences, as of yesterday, it was 63 million. The universe of all protein structures, 120,000. So essentially 120,000, 63 million. We have experimental structures for around about 13%. That number is not quite the translation of what we say, because this is an estimate. So the actual number is smaller. When we do whatever I just said, so we copy the coordinates through a simple sequence alignment, then we cover this 22% on top of whatever we have. Then when we do the profiled sequence alignment, then we get to round about somewhere this point. And this is the fraction for which we can apply this simple idea and predict the 3D structure. So essentially we can leverage the 120,000 structures that exist. We can use them as a template for over half of the 63 million, right? For say almost 40 million. Is that clear? It also means that for the other sort of, in this particular case, around about 40% or roughly half, we can actually not do this. And that other half is what the lectures from now on will target. But it is important to say that the simplest process that you immediately guessed, or I don't know whether you knew it before, comparative modeling, that the simplest method already captures half of all the pi. And for the remainder, I'm going to give you a lot of complicated tools that don't get very far at all. So the simplest step is reaching very far and is very, very accurate. And the details that follow up are still not getting us very far. But again, there's a lot of work that has been done, and I'm going to show you some of that over the course of this lecture. The story is slightly different when, rather than using this the tie being the entire universe of all sequences, we look at the universe of, for instance, one organism, here man, for instance, and then the numbers are a little bit lower. But okay, let's get back to the concepts. The word is comparative modeling, homology modeling ultimately is the same thing. You have one sequence and a structure known for and experimentally known. You have another one where you don't know it, and you find that the two sequences are similar, and you essentially model it. Target is the protein to model, template is the protein from which you model. And again, the first step is to identify the template. That you do that by simple sequence alignment. So you take your sequence, align it against the entire protein database, and find the one that is most similar. If you don't find any, end of story. If you do find one, then the methods that I will talk about today will begin. So we find a template, we build a model. Once we have built the model, and this is something that was also said here, the next step would be to assess it. There is the statement before, assess the probability of a fit from sequence. That's one assessment, but that's the assessment of this part here. Now we have a 3D structure. And the 3D structure may ultimately suggest to you that you need changes. How could you imagine this? So you build the model by the simplest approach, simply copying the coordinates. What could you assess here? What could you see when you build a model that tells you something has gone wrong? Can you imagine something? The simplest thing. Or is there anything coming to your mind? Let's not talk about a complicated energy function. Let's just look at something more visual, simple. Anything? So again, you take a sequence. You find another protein of a similar sequence. You begin with a human protein. You have the structure for the fly protein, for instance, the purple one in the previous structure that I showed you. You take simply for every corresponding residue, corresponding means you align the two, and then you find which residues correspond to each other. You build a model. You copy the coordinates. What could go wrong? I mean, again, so you get the coordinates now for human protein. What kind of things could you see that immediately would worry you?
The models don't look alike?
The models don't look alike. They have to look alike if you copy the coordinates. Then if they do, this could be true. If the models do not look alike, find the bug in your copy. So essentially you cut and paste the coordinates. If the two don't look alike, this is a simple spot of a mistake in your program, right? What else could happen? And it's true, it could happen, yeah?
Maybe that, like, because of chemistry, two amino acids cannot match.
So it could be, it could be that we have two amino acids that don't match. But in fact, in your human protein, this model implies that you have to put two positive charges next to each other, which is something that you all know is energetically not very favorable. This could be one way. Yes, that would be one problem that you would have to correct, in that in assessing the model, then the score would be very bad. What else could you think about? Let me go back, and I should have arranged my slides differently, and just visualize the problem. So again, we have this alignment, the corresponding residues, and now you have these two structures. The problems that I talk about you will not see in this example, because in this example everything sort of fits. But what else could you see? Between the, now unfortunately I show again the, okay, I have to go back for the, I believe the strongest one was the purple and the green here. What else could you run into? What sort of problems could you run into? Again, you could have the purple may have a positive and a negative charge, and the green then would have at the equivalent positions positive and a positive charge, and that would not be good. What else can you think about? Nothing? Remember, residues or amino acids have different sizes.
Maybe even if part of the structure fits well.
Exactly.
The other part.
It doesn't fit well, and that could be a result of the size difference. So you could have a bunch of large residues, where in the original one you had small ones, and they would fit. But with the size of the larger ones, it just doesn't fit. Right? So you would in fact have to pack volume into a space, and you don't have the space for the volume in the model. So somehow that means you have to sort of expand. Here is an example, no, this is an extra helix. Here's an example where an expansion could go to, so you have an additional turn or something like that. That would be an example for that. An expansion could be, let me find another, no. Yeah, so here the helix is a little bit more bulged out, takes a little bit more space, is a little bit wider. Again, this could be pushed. And that again, you would, in your first score in the model assessment, you would then say the way it looks is not good. Whether that allows you to make the next move and put it further is a different question, because that again depends on, is there something else? It may, if you put it out, is it going to clash with something there, right? But in the first step, really, you assess the model that you built, how well is it? What else could you think about? It's very related. You could have clashes. You could have essentially two amino acids sitting at almost the same, or at their side chains. So part of the amino acid sitting at the same space. It's related to the size problem. So that gives you a model score. And sorry for not having the right slides where they belong. Here's an example. Where in a particular case you would have to stretch, but you cannot stretch because the orange is there. So that means that the refinement, the next step here, is not that simple. So every single time you imply by the model score that something is wrong, and you try to correct it, it may create something else that is wrong. And so you essentially move step by step. You correct one problem that you have at a time, and you see how everything else fits then. And that is a complicated process for which there are different optimization problems. overall, comparative modeling obviously works very well if we have very similar sequences. I showed you this human-human, 85%. The limitation here if you have very high sequence identity is simply the speed of modeling. So the repetition of all those steps takes some time, and that is the major problem here. When sequence identity goes down, the quality of the model becomes the problem. So you have too many of these clashes. And you can, in the step process in which you sort of avoid these clashes, avoid these problems, you cannot solve them all. And don't find a good solution to sort of make everything fit, and that essentially leads to a highly inaccurate model. Further down here, the problem becomes the alignment problem, which means your alignment starts with the corresponding residues. Corresponding residues you align to proteins, and you say, well, that is the residue that I want to copy the coordinate from for this run, according to the sequence alignment. If that is wrong, you copy the wrong coordinates. And there's no way in which you can refine the model. Oh, this is not true. So today's methods find ways, but it's very complicated to then turn back and change the alignment according to the model. But that is, oh sorry, that's another problem. Further down here the problem really is, and that is the level where almost, so today the fly level we could track, but if you go even further below, you may simply not be able to see that these two proteins are related. Because you have to look at the sequence. And from sequence alone you may not recognize their similarity. That becomes the problem down here. I'm going to introduce two state-of-the-art models very quickly. And I'm just going to repeat essentially what I said so far for both of them, and they're both very similar. Both are distinguished because both have websites that actually put out 60 million models for the world to see. That is the Modeler website from Andreas Schali's lab at UCSF. And again, it starts with finding the fold, with aligning, and then building the model. Model evaluation, asking whether the model is okay, and then you go on two different paths. Either, if the model is not okay, you try whether there's another template. Is there another protein of known structure that may do the same thing in the first run already better? It may not be the one that you find first in terms of sequence alignment. It may not be the one that is most similar in sequence, but maybe it may be the one that gives you the better model. If not, try to change the alignment and go in some iteration and then try to improve the model. One issue here is the constraint satisfaction, and that ultimately is the point of bonds that you essentially, spatial constraints that you have to fulfill. Spatial constraints, the way you fulfill them, remember the background really, in this particular case, you have a distance between what is called C alpha. C alpha is essentially the main core of each amino acid, of each residue. And this is exactly what I said. They cannot be too close, they cannot be too far apart. You cannot just, you cannot bend. It's not a rubber band. They are very stiff. That means you somehow, this is essentially what a typical C alpha distance looks like. And that's what you have to put into your model. If your model doesn't satisfy that constraint, then the model is not good, and you have to introduce these movements. There is a sort of variable target, and this is sort of objective function. You try to reduce these constraints. You try to fit the constraints stepwise. That's what I said, and that is showing you how over time this can succeed. There's essentially it's a rigid body, molecular dynamics, so you cannot allow too much movement because then computational time explode too much. Another issue is you try to see not only is there another model that is best, but could I look at two different structures and take a little bit the best of both? So you try different models. Typical problems of this are what is called sidechain packing. So each amino acid is characterized by a core, so to speak, by the C alpha atom, and by something that is on top of it. That something on top of it is called the sidechain, and that makes out the different features. So amino acids differ by the sidechains. Bigger amino acids have bigger sidechains, and the sidechain packing may clash. So Other problems are really that you make essentially mistakes here. You misalign, I said that already, and you have completely the wrong template, and these two structures have nothing in common because you simply picked the wrong template. From the sequence match, it looked okay. The structure looks okay, it's just wrong. So sometimes you can essentially put a structure upon a sequence that sort of satisfies most constraints, but it's entirely wrong, and an example for that is shown here. That becomes then a challenge in molecular dynamics simulation to identify the best model. The best model in some cases, the best model is really the one with the lowest, so that is closest to the real structure. In some cases, actually it's not. So the highest energy model, the one that you favor in your simulation of molecular dynamics here, is the one that is in fact has a higher energy, but it is closer. So you don't find the right model. This is another problem. What now if the loop were missing? So this is another problem that I didn't talk about. The problem could be that a particular loop here is just not in your template. So you have the protein that you're modeling with, or that you want to predict the structure for. In this particular case, of my example, it was the human protein, the human KRAS, may just not have that loop, or may have an additional loop. What do you do then? Well, the first step is, essentially what you want to do is you want to find a loop that stretches from point A to B. You simply go into the database and look, do you find A to B in that distance, and pick all the loops that you can find in the database. You know something about the length of the loop that you want to span, because you know how many residues you have in addition, for instance, and you just pick the one from the database that fits best to your sequence, all right? Is that process clear? Anybody a question? How we can pick a loop from the database, and essentially you cut out a loop from the database and put it onto your protein. Because for that particular model that you built here, that one was, that loop was an additional thing that you didn't find in the known structure. So you copied from somewhere else. And this copy from somewhere else is no longer really sequence driven. Now you simply know, I have in my template, I have this point, I have that point, and the one that I want to predict the structure for, I have a long loop. So I have to find something that has, say this loop is 5 residues long here, that has to go from A to B with 5 residues. So pick everything in the database that goes from point A to B with 5 residues, yeah?
Could you maybe one more time understand what you mean by template?
Yes. So the template is so again, in the example that I talked about, I have a human protein for which I do not know the structure, and a fly protein for which I do know the structure. So the template in this particular case would be the purple fly protein. The target would be the human protein. I target this I want to predict the query. Let's go back to computer science, to language that you guys are maybe more familiar with, the query. So the target is the query. So I query, when the query is longer, so I have point A to B in the template being the fly protein, the structure that I know experimentally. And in my query, I have an additional loop of, say, 5 residues. So now I'm not going to take those 5 residues and do a sequence alignment against the protein database. Instead, what I'm going to do is I'm going to pick 2 points with similar sort of coordinates, or similar relations in 3D, and just find every single case of 5 residues that connect them that is in a loop. So I assume it's not a beta strand, it's not a helix, it's something that is sort of a loop. And I simply find them all, and I see which one fits best. And that one is the one I copy for my structure. Is the idea clear? Yeah?
Did you search for the loop for the human protein?
Yes. So in this particular case, in my example case, the query is the human protein, and I look in the protein database for loops, but I look for loops in all proteins. I want to model my human protein. But the loop that I copy could be from, bless you, could be from a bacterial protein, and in particular it could be from a protein that is completely different from KRAS.
Why don't you just take the loop from the...
So in this particular, so this is a good point. I'm sorry. Let me repeat this. The question is, why don't I just take the loop from the fly protein? The assumption is the fly protein doesn't have that loop. The assumption is that this is something that is an embellishment, an addition for the human protein. And typically that has a functional reason. But something that where the human and fly protein differ in sequence already, so something where in fact I cannot find in the sequence alignment the corresponding residue. In terms of sequence alignment, that would be an insertion. So this would be the situation where in my alignment I would say in fly F, L, Y, Y, L, F or something like that, and in the human I would have H, L, so I did a mistake here. And I would have 5 more residues squeezed in, and that would be A, C, D, let's just go the alphabet. Those 5 residues, they would be squeezed in there. This I can look up the structure for. This I can look up the structure for. Let's have the tryptophan here. But these 5 I do not find in the fly because they are additional residues, right? And in my alignment I would just have 5 deletions here or 5 insertions here. And again, the way I do this, I know the coordinate of this, I know the coordinate of that, and I look at relative coordinates that are similar, and I look for all the loops that span 5 residues. So that gives me, say, 50,000 or 20,000 loops. And then I ask, of these 20,000, what is best fitting to this sequence? And that simply I do by putting the sequences in the model and see whether there are any clashes. Yeah?
So what do you mean with a loop?
Why do I want a loop?
What do you mean with a loop?
So yeah, it's a misnomer. But what I mean by loop is this again here is the fly protein. This is the human protein. This is the query. This is the template. This is known 3D structure here. This is what I do not know. In the human protein for which I do not know the structure, for which I want to predict the structure, I have 5 additional residues. I just call them loop. Because the idea of comparative modeling is that essentially you saw all of these, that human and fly look very, very, very similar here, and then maybe they go into the next helix here, and maybe one of them has an additional 5 residues here. And very often that is in a region that is not a regular secondary structure, it's not a strand or helix. Very often this is something that is sort of loop, an addition. Yeah?
So are you looking in the database for those 5 residues, or do you look for those 5 residues containing fragments?
I did not understand the first part of your question. Can you say that? I just didn't hear it.
Because you said like you look in the database for these 5 residues.
Yes.
But just for the 5 residues, like just like
Oh no, so I'm not looking in the database for A, C, D, E, F. I'm now going into the database of known structures. And I'm picking all 3-dimensional objects that span point, this is one point and that's the other. Now I have 5 additional ones. And I have to go from that point to that one with 5 additional residues. I pick them all, say 50,000. And then I find the one that energetically fits, one way of doing that energetically fits best to these 5 residues that I actually have, these 5 amino acids that I have in my case. Idea clear?
How do we decide which one fits the best? Because we have around 20 to 50,000 results
Yes.
for a single query, and that's a lot of results.
Yes.
How do you determine which is the best?
So the question is, thank you for picking this up. I said, yeah, maybe I find 50,000. It's a big number, he says. How do I find the one that fits best? So first of all, I'm going to cluster them. I'm going to ask, are they all different? So maybe that already boils it down. But then maybe it boils it down to 5,000. So we still have a large number. How do I find the one in the 5,000 that is the right one? And my statement was, I'm going to compile an energy, and I'm going to hope that in my energy, somehow we are in a situation like the one that I indicated here. So this is thousands of different molecular dynamics steps for a particular model. And I hope that the one that is the right one, that is best, simply has the lowest energy. And that's the one I pick. I'll get back in a minute to some other aspect of that. But in principle, that's the idea. Yeah?
When we pick the structure that corresponds to the part of the structure, do we care about the sequence or?
No. So again, sorry. But let me get back to what he's asking, and I was too fast in saying something. I gave you the wrong answer. So what he's asking is, when I pick my 50,000, 5,000, whatever I pick, do I at all look at the sequence? My fast answer was no, because that's what I said before. So there are methods that for which the answer is yes, they care for that too. The problem is finding short motifs is very complicated from the sequence. So if you have 5 identical residues, they may look in structure very different. So even identity does not necessarily help you. So you hope that but then at some point you're at the end of the story and you have to pick one. So there are different ways of doing that. Some methods care, so implicitly for that, but in principle the idea is to not care for that at all and just look at the energetically most favorable conformation, because we have a 3D problem. You care for the sequence because the energy is defined, how well does it fit to that sequence. In that sense you care, but you don't care what the one you copied it from. Or again, some people do, or some methods do, but in principle the idea is not really depending on that. That would be a refinement step. Yeah, 2 questions, yes?
We make this assumption because we assume that signal structures have a sequence, right?
Yes, that's a very, very, very good point. And I'll get back to that point in a minute. So how come we can get out into the database? Doesn't that imply that we assume that all the structures are somehow in there already? And that I in fact can find a loop? Yes, that is indeed the assumption here. And indeed, we believe today that the database of 120,000 structures somehow has most important motifs, let's call them motifs, sort of 3D fivemers in some sense. We believe most of them are in there. We may not recognize them, so many methods try to sort of chop the protein into segments, chop the entire database into segments and find the right segments. Jigsaw is one of the methods that does that. And that somehow works, but not really. So we cannot find the right one, but we still believe the right one is there. Okay, in many cases we do, and in the next slide I show you what if not. But yes.
Maybe that's a stupid question, but
there is no stupid question.
The question of them before. Why is the low energy important for picking?
So let me repeat 2 parts of the question. The first was not a question, it was a statement, or an incorrect statement. And that was the statement of maybe this is a stupid question. There is no such thing, okay? Please understand that. I make mistakes in explaining things. If whatever I tell you is not clear to you, then that's my mistake. There is no stupid question. So the second part of the question is, I forgot the second part of the question. Ooh. Why is the energy assumed to be, why do we assume that the correct conformation is the one of lowest energy? Yes. Now we get into something that we believe. And there is an immense body of work in trying to prove that this is in fact true. And the way I see this is we assume that it is true, and it helps us to assume that. The assumption is that when you take a protein, you throw it into a solvent, that what actually happens is that the structure that is stable, that the structure that the protein adopts, is the energetically most favorable solution. So it can sample a large conformational space, so there are many, many, many possible structures is what that means. And the one that is actually native, that the protein normally adopts, is the one of lowest energy in all of these possible conformations in the entire space. That is an assumption. And that is the assumption here. There are two assumptions underneath my loop modeling here. First, that we have a measure of the energy function, which in fact we don't. So we have an approximation. And the second assumption is that according to that approximation, the lowest energy would tell. And again, I showed you an example of a MD model here, where the lowest, the one closest, so that's an RMSD here, so that means lower means closer in 3D structure. And the lowest in energy here is not the one that is closest. Closest are these, and they're very high in energy. So this is a typical example for it can go wrong, this assumption. Here's an example for it goes right. Here's an example for it goes wrong. But somehow it is also true that the group of the closer ones are also so there is no point up here. Okay, it's not that we have a very high energy and are very close. So the focus of these curves is such that the mass of the points down here is close. But yes, it's an assumption, and this is immediately showing how this assumption can be incorrect. Again, yes?
I did not understand the analogy with when I put the protein into salt, then I get a low or high energy. What do you mean by this?
So first of all, I didn't say salt, protein into solvent. Solvent is water.
Okay.
Is that answering your question?
No, I don't.
Okay, so then.
What do you mean when that it's low energy or high energy?
Okay, so what I said is you take the stretched out chain, the protein sequence, you throw it into a solvent, water. This is essentially the native environment in a protein in a cell. And in this cell, the protein will fold up. Fold up means it will adopt a lower volume. I said that some way of describing a protein sequence would be to say there are hydrophobic and hydrophilic residues. Hydrophilic means they want to be close to water. Hydrophobic want to be away from water. And you immediately see that if you somehow spread randomly or in some way the feature hydrophobic, not hydrophobic onto a chain, then you will create an object that will sort of adopt a smaller volume. Because whatever is H doesn't want to be close to water. So it wants to go into the inside. And that means there will be some sort of collapse. Again, you have a stretched out chain. Collapse means some residues come together in space. And this collapse is collapse because you have a reduced volume. And the reduced volume reduces the number of mistakes of having hydrophobic residues that hate water next to water. That's an energetic mistake. So in the simplest model of hydrophobic, non-hydrophobic residues, you immediately understand, just a sec, you understand how the whole thing collapses and adopts a situation in which you have the highest number of hydrophobic residues inside, non-hydrophobic residues outside. And that would be called the energy minimization in some sense. And this energy, the simplest energy here, it's absolutely not biophysical, is one where you say hydrophobic inside, hydrophilic outside. Hydrophilic wants to be to water, and you simply optimize the number of agreements to the simple energy function, 1 and 0. And again, the native protein folds in a way, collapses, takes a small volume. That corresponds exactly to, most of the case, or the assumption is, to the lowest energy. And the lowest energy, now not in the model of hydrophobic, non-hydrophobic, but in a more complicated energy model. There's another question before I go on.
How can we say that due to hydrophilic portion present in the motif, they will shrink? There is also hydrophilic portion.
Yes. Now again, so how can we say that due to, so again, my simplest model, I have hydrophobic and hydrophilic, and in principle I can in fact do that on a computer. And I can do a simple folding simulations. They do exist. Again, most likely on the Rosalind website, possibly on the Rosalind website or on other websites. How can I say that just based on that I will create a situation in which I will fold? Well, the point is, again, just imagine it as a computer optimization problem. You have mismatches to the energy function, where the energy function is so let's, in the simplest model here, you have H, hydrophobic, what can we do it? O, let's call them O, hydrophobic, and philic I. So you have O and I's, you distribute O and I's randomly. And now you want to create contacts such that ultimately you have all the O's sitting somehow together and all the I's sort of sitting around. Now you have the constraint of a chain. You can clearly not find the optimal solution. But whatever chain you create, you can find that one that is most optimal for this chain you have. And that is what would be called the lowest energy in this particular case. And you will see that no matter what sequence you create, the only way in order to optimize is to somehow put the O's together. O's together means contact. Contact means you increase the volume that a stretched out sequence would take. And again, when you boil an egg, a hard-boiled egg is just the opposite of that. By boiling an egg, you unfold the protein. By unfolding it, it gets bigger. So this is condensed, compact. Unfolding means making it like the stretched out chain bigger, and that's why it is, the hard-boiled egg is hard. Because it occupies more volume. Okay? As simple as that. The raw egg is fluid because it needs less volume. You pull out the protein into its original chain and you aggregate. I mean, you get bigger, you fill the entire volume, you aggregate and become a solid state, an egg. That part of the problem is clear?
So is the egg, when it's unfolded, is it high or low energy?
So the assumption is that the egg unfolded, so the boiled egg has a very high energy. So it's an unlikely situation. Again, this analogy, now let's be careful with this analogy. Everything we say moves toward lower energy, but the egg is not getting folded again easily, because when you break it, so then it aggregates in a way in which you cannot go back. For proteins, when you don't go to the state of the hard-boiled egg, in between there's a state where a protein can sort of unfold and refold, unfold and refold. In an egg, you're beyond that state, because it creates an aggregation of many unfolded proteins. So the egg will not spontaneously, in that sense, the image is a little bit misleading, that the egg, yes, the egg is the higher energy, but it will not just let the hard-boiled egg sit there for 50 years and then, yeah, well, then it becomes something else. But that's a different story. I believe this is clear. Yeah?
During this folding process, there may be charges present. They can repel each other, like charges repel each other. So how can you say that this structure will be stable?
So again, the statement here is, okay, in a real universe where we don't have hydrophobic and not hydrophobic, but where we have a little bit more complicated energetic situation, we may create a situation in which we have positive and negative charges, and they may come close to each other. So how can I say that the resulting structure will be in fact stable? And the answer, again, is the same that I said before. You put all of these features into an energy function. And that, in fact, is what nature optimizes. All of these forces, the biophysical forces that make residues be happy in the environment with each other's charge, with water, or whatever is next to it. And the solution, the native protein that is stable, is the one of lowest energy that satisfies all these constraints best. And again, we can, in principle, do that in a computer. The problem is we cannot get to the time scales of folding, and we can do that for 5 residues. And again, I'm getting to that issue in a moment. But we cannot quite simulate a protein structure. Again, there are computers out there that can do it for small proteins. There's one computer out there, so that sort of beats the world by a factor of all the academic world by a factor of 10. It's called Anton, and is in New York. But David Shaw this company, or Anton 2, whatever it is. It's a specialized parallel machine that does the molecular dynamic simulation. In fact, puts all of these energy functions into a computer and simulates how the protein looks, adopts a lowest energy function, and in fact shows that we can do that for proteins of 50 residues on a time scale that is very close to how folding happens. And again, this is a factor of 10 faster than anything that exists anywhere else in the world. But again, so we can still not, even that, so that's the top we can do, and even that cannot really simulate typical proteins. Some aspects of it, and this is really the breakthrough of the last decade in that sense. But again, so I believe I back to the story, the loop modeling, everybody's clear about that, is that right? So now that gets us to the next step. What if, yes, the assumption was that the loop I can find in the database. What if I cannot find that? What if that particular loop, I find nothing in the database, or nothing that is somehow energetically favorable? What can I do? And the answer is exactly what I said before. I can do the molecular dynamics. I can simply put this into energy functions and just simulate it in the computer. I said this is not possible for very long proteins, or for typical proteins, but for 5, 7, 8 residue loops, in principle it should be possible. And what this graph shows you is the loop length from 0 to 14. I cannot remember why there are two graphs, and it shows the mistake that you make, so the increasing mistake. If the loop is one residue, you get it right. And then the mistake goes up, and at some point, it's difficult to say where that happens, but most likely around 10. The mistake that you make is so high that you essentially cannot distinguish between a loop that looks like this and a loop that looks like that. I do not mean to have two mirror images. So what I'm trying to say here is you cannot distinguish between the right solution and something that is completely incorrect and has a very different orientation from the right solution. Now that is important for the functional interpretation. So ultimately that means we can actually, today we can push this a little bit further. This is not the most recent publication, we can push it further. But still, this gives you some idea. We cannot model a loop of 20 residues. So the only reason why Anton can handle 50 residues is because there are more constraints than typically in a loop. Okay. Here is another model that also is on the Swiss model, is also in the internet. And the idea of Swiss model, there were two important ideas here. To have something that's fully automated, that is for not expert users, and then there was this part here. And that was after over a decade of research, experts in the field said, do less, because if you do less, you make fewer mistakes. Everything we have learned over the last 10 years, they argued, proved us that we don't quite understand the problem, so the less we do, the better. And one interpretation of that is the original algorithm was you simply do the alignment, you run your sequence against your query against the protein database, you copy the coordinates, end. No model assessment, no correction of clashes. Leave it to the user to realize something is wrong here. But ultimately, as a method developer, that's the best you can do, as long as you want to do it completely automated. And that, for many years, was, in fact, surprisingly, one of the more successful methods. Because for many years, people had done a lot of intelligent work on top of that, and it had not really worked very well. We have learned over the last 15 years, or 20 years, and today Swiss model is way, way more complex than that. It goes into a similar circle cycle as the one that I showed you from Modeler, but let's just explain some of the steps. The first one is that it uses better ways of alignments, sequence profile steps. It uses predictions of secondary structure transmembrane EDCs that we will get to next week. It builds a model core. So first of all, again, it finds the template, builds a model core, does the loop story that I talked about, sort of tries to energetically place side chains correctly, tries to energy minimize. So this is again, once we do have an understanding of the biophysical energies, once we are close to the right solution, or we believe we are close to the right solution, we can possibly minimize the energy. And today, in Swiss model, after many, many, many steps, this is the kind of model that you get. And these models have, in many cases, are amazingly accurate, but even in those models, so this is the sequence of the protein. In this particular case has 350 residues. And essentially what this one shows here is the difference in happiness of the residue. And you see that some residues, so all you need to see is 0, would be, that model would satisfy all energies. And that is absolutely not what you see. You see a variety of different features plotted here, but you absolutely see that none of them is 0 full stop. So the final model that you have, in fact, is clashing with many of your concerns about energy. Okay? And that is then, at the end of the modeling, the best you can do. You cannot refine from there, because this is the end product. You reduce the energy as much as you could, and you still see certain problems. And then all you can do is sort of point to those problems and say, well, this is all we can do. Now, this brings us to Cécile Parin-Pipe from René Magritte. You may know this image here. So this is not a pipe, is what it says. Everybody understands why.
Not a real pipe, it's a visualization.
It's an image of a pipe. And an image of a pipe is not a pipe. An image of a pipe, and we're back to image and reality. An image is something that helps you to visualize something, that helps you to make certain decisions. But maybe your experiences are such that you can even remember the scent. So you see this image of a pipe and you begin to smell tobacco. Or somebody runs out because now they have to smoke. I don't know. But the important thing is to realize this is not reality. And that brings us to the point you have to realize that the model must be wrong. That's the meaning of the word model. Okay? Intrinsically. And one way of best working on that is to really look at the model and see where is it wrong. Whenever you do develop a method that predicts something, try to come to the point where you identify what is wrong. Tell the user, this, okay, I'm going to give you the right prediction of whatever it is. Who's going to be the next winner at the Oscars or something like that. And then give a probability for your prediction and give some arguments why certain things are wrong and where things go wrong. And maybe the user can add that up and build a better model. Or maybe the user can simply zoom into the regions that are more likely to be right. Or maybe from the clashes you learn something about the function of the human protein. Ultimately, here's the statement from Thorsten Schwede, who developed, or is currently on top of this model. It is essentially a tool that allows you to do an experiment. It allows you to do the next step. And when you develop this method as a programmer, or methods, whatever method you develop, think about them also as tools, not only as end products. Okay? As something that people interact with. Okay. At that point, I'm going to go into a quick run through something else, which is how can we predict protein structures and how can we actually do experimental structures. The first part here is the protein structure prediction problem is solved. I'm quoting 3 different time points at which in 2 different journals this headline was published. The protein structure prediction problem is simply the problem of taking the chain and predicting how the 3-dimensional structure works. Okay, so all 3 clash with what I told you. So you being nice to me, believe me. So you believe that these articles are wrong. But even if I hadn't said that to you, one thing is obvious: you cannot solve the same problem 3 times. So if that article had been wrong, right, then that clearly was wrong. Now, what goes wrong? And what goes wrong essentially that people take some toy problem and they overfit the parameters. So they use the biochemical energies or biophysical potentials that they have, and they fold one particular problem for which they know the 3D structure, and they say inbuilt into their model is everything they know about that structure, and essentially they overfit the model. Right? And then they reproduce the model and then they publish, we solved the problem. How could you avoid that? So another thing is, when science goes into journals such as New York Times or Washington Post, that means that we have a problem that really has high attention. So many people care for that. And even on that level, people make obviously mistakes. So how do you believe it happens? Or how could you avoid this mistake? It's not because they didn't try to avoid these mistakes. How could you avoid it? Believing that you solved the problem by using some energy function. Yeah? So peer review. Yes, I have to I have to be a little more careful here. Typically, it makes newspapers like that only after it had been peer reviewed, published in Nature and Science. So this was underneath. There were peer reviewed. These all were peer-reviewed publications that then went on. So peer review is in fact what we try in science to avoid mistakes like that. And in these cases, and in many other cases, it didn't work. And it didn't work because the peer reviewer just looked at the structure that was right. They looked at the model, compared these two things, and they were right. They could, in their computer, in those days it was not really their desktop computer, but they could essentially apply that model and would get the same result. So everything looked right. Yet it was wrong, because it was overfit. How could you avoid this problem of overfitting?
No, about the right and wrong, like the theory is right until it's proven wrong. So in the 60s it was right because like...
No, so yes, the statement here, I want to repeat that because it's a very important, valid statement. Theory or model or statement is right until proven wrong. So the statement now is in the 60s maybe it was right and then it was wrong. No, the reason why it was later found in the 60s found to be wrong is that it didn't work for another protein. It worked for one particular protein, full stop. In the 60s, we had only, and I'll get to that in a moment, we had only 2 proteins. So, and I will show you something about those 2. I will show you those 2 proteins. But in the 70s, we had more, but we still had a very limited number. In the 90s, when the last one here, the Washington Post article came, we had almost 100. But still, it was not easy to really trap that. Peer review had not trapped that. So at that point in the 90s, everybody was scared and had seen the other articles before. And many who had seen that felt like something went wrong. But I didn't know what had been gone wrong on this one, on the last one here. I clearly remember that story. I had the sense it couldn't be right, but absolutely scientifically, I could not argue what was wrong. And ultimately, that was proven wrong by the answer to this question. How could you assess prediction performance? Any idea? And again, this is essentially something that it's valid for protein structure, and I'm talking about an experiment that was introduced in the field of protein structure prediction, but it's something that you need in any data mining. And the more we are doing, we are developing methods that are relevant for society, for economy, and the more under time pressure we are, the more it becomes urgent that we really find ways to do something like this, although this involves a time frame that is typically not realistic for you. But essentially, what's the idea? What can you do?
You can predict for something that you know the answer.
For something that you do not know the answer?
No, you do know the answer.
Yeah, but that's what they did. So they predicted for something for which they know the answer and they overfit the model.
Trying to find a new one.
Sorry?
Trying to find or trying to predict from a new sequence.
Trying to predict?
From a new sequence.
From any?
From a new sequence.
Ah.
Trying to find a new protein somehow and it's like, I mean.
But again, so here's the problem. Here's the intrinsic problem. Either I try to predict something that I already know, and then I have the problem of overfitting. Or I predict something that I do not know. But then am I right or wrong?
Cross-validation?
In principle, that's the important answer. This is not what I'm going to show you next, but in principle for machine learning, the important answer is cross-validation. So you pretend that something is not known. So you have 100 proteins, and you take 10 out, hide them under the table, and you pretend you don't know them. And over the course of the lecture, we will get back to this. This is the most essential part of machine learning, and this is where usually machine learning goes in some sense wrong, because it's very difficult to do that right. But let's step back and let's assume we are not in the world of machine learning, but in a world where something else happens. Here's an idea. It's the CASP idea. The CASP idea is to in fact predict for something that nobody knows at this point, but that we know will be known soon. And that is again what I meant by time frame that is not quite right. So one way, so this particular experiment works the following way. April to May, the organizers collect cases of proteins for which over the next few months, some experimentalists will do the structure. It's not known today, but we have talked to the expert and they told us that they will have the 3D structure soon. Okay? Then methods predict over some time. Then all these predictions are assessed, and then there is a meeting of coming together. Over time, the CASP is still running. It was running every 2 years. This is the number of groups as it exploded over the years. And one important finding of CASP was that the only thing that really works is this comparative modeling that I talked about. That in fact there is no prediction from 3D structure from sequence yet, and that there are many improvements on small details. One important effect of the CASP competition was that people started doing service. So whatever worked in some lab, somebody else used. And people collected, and the world of structure prediction became communication between meta and meta and meta, and so forth servers. So again, I go into 3D structure determination now. This is not something that we can do on every master thesis or something like that. But sometimes on the master thesis, one way in which you can do that in cross-validation is you begin your master thesis today. You collect your data essentially over the next week or whatever time frame. And the timestamp on the data is that week or whenever you begin your method. Then typically it takes a few months, say 5 months to develop the method. At the end you have a model, machine learning model. Now at the end you can just go back and see over those 5 months what has been added to the database. You have never seen that before. And over all the things that have been added to the database that you had never seen, that even a cross-validation you had not seen, how well does your method do? And in most fields something like that is possible. And in that sense you can copy the CASP idea for most of the work that you will do in terms of machine learning data mining. Let's talk about how we get to 3D structures. The one aspect again is that there's structures on different levels of details. On the left what you see is a cartoon that highlights the secondary structure, helices in red, strands in yellow. By experimental structure determination, so in terms of numbers, and this is not the latest version, I actually don't have a date on here, but most of them come of the PDB structures, then were 84,000, most of those from X-ray crystallography. X-ray crystallography, it begins by taking the protein and getting a lot of it, expressing it, purifying it, and growing a crystal. That's not easy. It takes a long time. Once you have a crystal, you shoot X-ray beams onto it. And those X-ray beams come from a synchrotron. They have to be highly focused beams. And these beams are diffracted. And from the diffraction pattern, you get an electron density map that gives you a probability where the electrons sit. electron density map already gives you an idea that what you get is not a coordinate. You get a probability. Okay? And then into this electron density map, you have to sort of thread to fit in your chain, your sequence, your protein. So you have to then essentially guess how the actual sequence or the actual protein that you have put into the crystal fits into this electron density map best. And then by fitting you get an atomic model. Atomic model essentially means that for every single atom in that protein structure, you have a model where it sits. Not only for every C alpha in an amino acid, you actually every single atom. And one that is shown here is myoglobin, and myoglobin in fact was the first structure in 58. Okay, another way of doing it is by NMR spectroscopy. The advantage of this is you now can put the protein in a solvent, in its native normal life condition, into a little test tube, put that into a big magnet, and simply excite the protons and see how they relax, the spins relax, and from that you sort of see the interaction between residues, you see contacts. The problem here is the scale of the magnet, or the challenge somehow. This is City College, full of a bunch of machines like that. So the person on this, so the magnet, this is the person. This is only a magnet. There's this, the size of this thing is not explained by anything other than that it's a huge magnet. It's the kind of magnet that if you are anywhere in this room with your credit card, you no longer have credit problems with that card, it's dead. And anything else you carry that is magnetic that comes anywhere near, so there's a Faraday cage. The whole room here is a room as big as this. So anything that sort of you want to keep safe, you have to keep outside. The problem really here in this particular case, the problem in the other one, the problem was to get the crystals ultimately. The problem in this one is to actually really determine the structure, because it's a very tedious process, and you can only do that for relatively short proteins. Short proteins, 200 residues. I told you most proteins on average sort of is 400. So this is the subset of shorter proteins that you can handle with that. And ultimately what you get is a bunch of models shown here. So every one of these traces here is one different model. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and so forth. You get the idea. And you see that for some regions here, all of these 15 or 20 are at the same place, and for other ones, they're completely different. The interpretation is, so these different models are simply different ways that you can fit your structure to the constraints that you actually measure. What you measure, again, are essentially distances between protons. And different computer solutions, those are all models built in the computer according to the experimental constraints. Different solutions fit to the same constraints. And the interpretation is that those, in fact, where they're all the same is where the protein doesn't move. And those where your constraint fit different models is where you have variability, movement in the protein that has to do with its function. That's the positive interpretation of this image here. The negative interpretation is those are your errors. Your experimental constraints are just not good enough to define exactly the right model. And that essentially is an error in the process or the procedure. NMR is still relatively small. It's about 10%. The number here is increasing, but not that much. Actually, the relative ratio between X-ray crystallography and NMR stays pretty constant, because NMR is complicated. But NMR allows you to do things and see mobility that X-ray crystallography doesn't quite. Here's another story. I told you it's complicated to just look at a protein structure by microscope. Here is a technique by which you can do that, cryo-electron microscope. So you freeze the protein, not a crystal, it's not its native condition, but at least you don't have to for a long time, put it in conditions under which it crystallizes, it's still native, and then you do electron microscopy. The problem is now that you very often don't get a high resolution. So on a resolution of 32 angstrom, 16 angstrom, 8 angstrom, 4 angstrom, and essentially this is all you can resolve, this is all the detail you can see on the 32 angstrom. Most EM in the database are somewhere in between these two. Today we have techniques that go, or not today, the latest techniques that now people invest into go below this. And maybe if I gave this lecture in 5 years from now, I would say that most structures really come from microscopy. This is a technology that is evolving rapidly, or maybe 10 years from now. But this may really be the future. At this point, in most cases, the resolution is not good enough to give you atoms, or to give you details that you need for drug design, for instance. But you can combine modeling and EM in a way, for instance, that you see, if you see these blobs here, then you can underneath see which protein structures you know by comparative modeling that you can fit into these blobs. Okay? And so you can hope that the blobs give you constraints for fitting in certain models that you already know. The more we cover by comparative modeling, the more of these original elements of these blobs you will already know, and the more likely you will be able to somehow connect them in a way that fits to these blobs. And that again is a technology that works very well. There's this, yes?
What does the percentage mean?
This means the percentage of the entire, oh, this is of course completely wrong, 100. So this is 1% of the entire database at the time. So 1% of all the structures that we know are EM, electron microscopy. 10% are NMR, 89% are from X-ray crystallography. Sorry, there's a mistake here. Resolution, just what I show you here, different resolution. So the green here is the real structure. And the blue is the actually underlying experimental data. Now you see that the green here, the blue, you could move the green around. This is 3 angstrom. 2.7 angstrom, 2 angstrom, 1 angstrom. Now in 2 angstrom you see that the blue is actually already beginning to show you the ring. So now you can maybe flip this a little bit, but there are not many conformations that are compatible with this blue here. And here, the conformation in the blue, again, I'm sorry, I would have to completely darken the room. You even see dark in the middle. So the blue even confines, it sees in the center of this blue. It sees beyond the actual atom in that sense. Into the atom, really, beyond the molecule, in great detail. Very, very, very few structures are like that. Many are like this. That's the sort of level that you get, so you have some interpretation in all the structures that you know. Okay, now on this model from 3D, 2D, 1D. 1D, what I show here is secondary structure, and they are stabilized by hydrogen bonds. And this concept was introduced and given a Nobel Prize to by Linus Pauling in '51. So he predicted the existence of secondary structure of helices and strands before people had ever seen a protein structure. And he predicted this based on a very good understanding of the features of proteins. So there are 2 major elements of secondary structure, the helix. And the helix is stabilized by a hydrogen bond that forms between residue I and I plus 4. I and I plus, so it goes around the I and I plus 4. And creates a network of hydrogen bonds. They're relatively weak, but there are many of them, and they stabilize this helix. In contrast, a beta strand, you have the chain running here, and another chain, or part of the chain, running here, and they are, every second residue in this is binding to another one. So these two strands, that's one strand, another strand, form together a sheet, and these sheets are essentially stabilized by the hydrogen bonds of I and I plus 2. So, but again, so the binding partner here is I plus 2. The binding partner is further away. It's J and either J plus 2 or J minus 2. So it could be in that direction and that direction, right? Concept is clear. And those, in fact, are the two major elements. We have seen them. You have seen them before. Red helices. Those here, yellow, are the strands. Again, if you pack two strands together, you call it a sheet. And these are, in fact, often shown as arrows, where the arrow simply shows the orientation. So the advantage of the arrow is twofold. One, you see whether they are, in particular in this particular case here, we see both this and that are sort of parallel, and these two are anti-parallel, so they are in a different direction. These are parallel. And from this image of the extension, somehow the paper kind of thing, you see the orientation, roughly, of the sheet. Again, I said that the first protein structures were determined in the 60s. John Kendrew got a Nobel Prize on myoglobin and hemoglobin. Max Rost got a Nobel Prize. And they looked like this. Those were the first protein structures. And actually for a long time, the only ones. What do you notice?
Most of them are helices?
Yes. They don't have strands. So essentially what people saw in the first structures were helices. And that was a clear conclusion that there are only helical. And that Pauling and his colleagues have been wrong. We have only helices. And for many years that's what people really believed, because that was the only data they had. The third protein was also an all-alpha protein in the 60s. Now, how can we assign secondary structure from 3D coordinates? So I give you a set of 3D coordinates. How can we find secondary structure? There are two main methods that I want to briefly mention here. The first one is define in DSSP. In define, essentially, we go through that. Define essentially looks at the geometrical features. You see things that come together, you see things that sort of do look like that. And from these geometrical features, you define secondary structure. DSSP uses hydrogen bonds, just like Pauling predicted the formation of beta strands and helices. So it begins by simply compiling whatever is a hydrogen bond by a simple, totally simple energy potential. Electrostatics potential, and then with this simple potential you simply assign all the hydrogen bonds, and then you simply do I and I plus 4. If you see I and I plus 4 continued over the sequence, you call it a helix. If you see the I and I plus 2 pointing in a hydrogen bond to another, J plus 2 and J minus 2, you call it a sheet. As simple as that. This way you can simply get from 3D coordinates secondary structure assignments. And those secondary structure assignments we will talk about in the following lectures, because we're trying to predict secondary structure. There are different methods of assigning secondary structure. The most successful one, and essentially the only one that still everybody uses, DSSP. And they agree to only 30% of all their assignments. With that, I'll leave it for a few questions, but that I will, with that, close lecture today.